Thank you, Christa. Well, hello, and thank you for coming to my talk, everybody. Yes, it's not a very inspiring title. I did think that once I'd sent that in and had my abstract accepted. But really, it's about competition in plant communities and how we measure that. Maybe that's a slightly better title. My name's Lindsay Turnbull. This work, of course, is a collaboration with several other people. Sean Tuck was a PhD student at the University of Oxford who finished in the summer and sadly went off to do other things. Janielle Porter was a PhD student at the University of Zurich when I was still working there. And Mark Rees is a long-time collaborator. He's also a plant ecologist, and he's based at the University of Sheffield. So this is a piece of work that we were all involved with. And I want to start, and my picture there is just of a diverse plant community. I call myself a plant community ecologist, and that's what I mean by a plant community. It's just a group of species growing together in the same place, basically. That's all we mean by that term. And you can see that this plant community happens to be a rather attractive one. It's a nice meadow in Switzerland. There are lots of different species present there. And one of the most obvious ones is that nice scabious there. It's probably Knautia, and it's that lovely lilac colour. And I guess what I'm interested in is if I went to that meadow and I removed that species, I took that Knautia or Knautia, depending on what part of the world you come from, I took it away, what would happen? What would be the response of the species that are left there? There's two options, really. They might expand, increase their population sizes, use up the space and the resources that were left, were being used by the Knautia and are presumably free now. And the other species might fill that Knautia-shaped hole that has been left in this community. Well, the other option is that they don't, and there remains a Knautia-shaped hole in that community. And I'd just like a quick show of hands, actually. If you'd like to just vote on those two options, I'd be interested to see what people think. So who thinks, or could you just raise your hand, please, if you think that if I remove Knautia from that community, the remaining species would respond and fill the gap left by that species? Thank you. And now, could you raise your hand if you don't think that will happen? If you think if I remove Knautia from that community, a Knautia-shaped hole will remain in that community. Okay. So it's a pretty overwhelming majority who believe the first of those statements, which is fine. It's interesting that it's not unanimous. And I think if you read the plant ecology literature, that's not necessarily surprising. We still argue about that, which in some ways seems odd because it seems like such a fundamental thing that we really ought to know by now after 100 years of doing ecological science. I guess I think the first, that if I remove that Knautia from that community, I will see a response by the other species. I think because I see plant communities as being fundamentally competitive, that these plants compete for the space and the resources in that community, and that they therefore suppress one another. And if I remove one of those species, I expect to see that competitive release. So the Knautia is there, is able to exist in this community because it is a good competitor, because it can make space for itself, it can access resources, and if I took it away, then other species would be relieved from a little bit of competitive pressure. Okay. But in a way that's, not a great understanding. What I don't really know is exactly who's going to respond. Are some species going to respond much more than others? Does Knautia suppress some species more than other species? Who's going to be able to take advantage of that? And to understand that better, we need to know more about the competitive interactions and how they work in a community like this. Who is competing with who? And that's something that we really struggle to get a handle on, to really be able to quantify competitive interactions. And of course, lots of competition experiments are done by plant ecologists, but a lot of them are done outside of real plant communities, and they often reveal that there are very strict competitive hierarchies among species. But of course, we don't know how to use that information either because it didn't take place in a real community, which is where we'd really like to measure these things. So here's one way of doing it. When I was doing my PhD, the fashion was to try to use observational methods. Instead of experimentally doing things, we would try to use the variation present in natural communities to quantify the strengths of interactions among species. And here's an example of using that method. Now, I would go to a community and I would nail down a quadrat. Of course, I'm a plant ecologist. I must use a quadrat. And this quadrat is divided into lots of small cells, and it's a permanent quadrat. It means I can come 1 year and I can count the numbers of each of the species that are there in each of my small squares, and I can come back the following year and I can do the same. And what I can see here, if I focus on this species with the solid round blob, that in this small square it went from one individual in year T, and the following year there was still one individual there. Now let's imagine these are annuals, so they're not exactly the same individuals, but nevertheless the population size didn't change. In the top right-hand quadrat we had one individual in year T, but in the following year there's two. In the bottom left, one became three, and in the bottom right, two individuals became none. And that gives me variation that I can work on. I can model the change in population size, the delta NT plus 1 NT, in each of these squares. And in some, the population growth rate is positive, in others it's negative. And what I'm hoping is I can use that information to pull out the impact of these different species on each of my focal species. So I could build a model where I could model the population change of each focal species as a function of the number and type of neighbours. And I might expect if in year T this species has a lot of neighbours, they will be suppressing it and suppressing its fecundity, and therefore you won't see such high population growth rates. All right, so this is one common method that plant ecologists use. Other methods involve neighbourhood modelling, where you have very detailed maps just from a single year, but you can effectively use the same idea. And people have done this for a whole variety of plant communities. And quite often what they find is something rather odd. They find that each species, each focal species, is strongly impacted by neighbours, by conspecific neighbours, by individuals of their own species, and that the strength of interspecific competition in these communities is generally rather weak. So it seems that they're not really being suppressed that heavily by individuals of other species, but they are being strongly suppressed by individuals of their own species. Now, of course, if you know about plant niches, maybe that doesn't seem terribly surprising, because we kind of know that's one of the conditions for coexistence, right? Intraspecific competition has to be stronger than inter. But still, it conflicts with a lot of experimental evidence, which says that actually some species are better competitors than others. And actually, if you then take this a step further, and there's a very nice paper by Martorell and Freckleton a couple of years back, they did exactly this in a desert community in Mexico. They parameterized their model, they estimated all their competition coefficients, and then they went a nice logical step further and said, well, let's use that model to see what will happen to each of my focal species when I remove competitors. What am I going to predict happens? And they predicted not much, actually. I mean, not much can happen if you're not predicting that there's very much interspecific competition in your community. So we're kind of left in a bit of a mess, aren't we? Because most of us agreed that if we removed scabies from this community, we would see a response by the other members of that community. But now when people go out and parameterize models of competition, they don't conclude that at all. They conclude the reverse, that each species is somehow a little island unto itself, and it's not really impacted by the other species that are there. So I think that's a bit of a conundrum that we need to solve. What's going on there? Is it that there's something wrong with these observational methods and not giving us the right answer? Or is it something more subtle that might be happening? And I realised I had an experiment going in Zurich at the time, and I could use data from that experiment. It wasn't set up for this purpose, to investigate this a little bit further. So here is a plot from that experiment. It was set up with a quadrat already permanently embedded in it. Okay, so this is a concrete lattice. It's filled with soil, a very sort of poor soil sand mixture, and 5 annual plants have been sown into it that come from sand dune type environments, and they were all initially sown with small numbers of each of those species, the same numbers, and then they were left to run for 4 years. So they're setting seed naturally, they're dispersing within the plots. There are barriers around here, so between the plots they can't disperse. So it's sort of a semi-natural system. And because of the nature of this, I can easily apply those methods I just said. So I can come 1 year and I can count the numbers of individuals of each species in each cell, and I can do that again next year, and I can fit some nice models and estimate the strength of competition for each of my focal species. And when we did that, this is what, so we did that first, and then we can do that second step and say, right, now I've done that, now I've got that model, and I've estimated the strength of interactions, I can predict what will happen to each of my focal species when I remove the competitors. And that's what we did, and that's what I'm going to show you now. So here are my 5 species. This is the smallest seeded species, and they get larger as they go down. And this is across all the cells, across many plots, the expected response when I remove all the competitors. And it's scaled to 1. So a value of 1 means absolutely no change, okay? A value of 2 means that the population size is predicted to double when I remove all the competitors in that cell. And a value of 8, for example, means I'm expecting the population to increase 8-fold when I remove all the other competitors from that cell. And these are the distributions, and it's not very remarkable, really, right? I'm not expecting to see very large changes, very large increases in population size when I remove all the rest of the competitors. So that's very similar to what Freckleton and Martorell found, and indeed what I'd find from several other papers if I bothered to take this next step and make these kinds of predictions from their models. But I don't have a test of this, right? I mean, the problem is, it's all very well predicting this, but I don't have anything to compare it to. But in my experiment, we did. Because at the same time as setting up these plots with all the species growing together, we set up monoculture plots. So we have got plots from which all the other species have been removed. And so I can actually test my prediction and ask, did I make a good prediction? And the answer is no, I didn't. I made a really bad prediction. So here are my 5 focal species. The 3 data points represent 3 different model forms. Obviously, there's lots of ways you can model this change in population size from year to year. You can make slightly different assumptions. You can use slightly different functional forms. But the fundamental answer is that all these models are bad. And I don't know that when I fitted the models, all the diagnostic plots from fitting the models were wonderful. It looked like I'd fitted a great model that explained a very large percentage of the variation in my data. But that doesn't matter. The model does not make a good prediction, okay? Can I just finish my talk and then you could ask me that? So here I have, so this would be a perfect prediction. This is, if I was right about if my prediction was correct, I would get my points settling around this line, okay? So what I'm doing for most species is massively under-predicting the effect of removing competitors. I predict that they won't respond much. In reality, they respond a lot more than that. They are experiencing a lot more interspecific competition than my model says they're experiencing. And in one example, it massively overpredicts the extent of competitive suppression by other species. Now, why might this be? I don't know. I can only speculate. But what we think might be going on here is that species become confined very quickly to the places in the environment where they actually compete best. So even in an experimental setup like that, there is underlying environmental heterogeneity that is extremely difficult to measure, and that's operating on a micro scale. And if species start to end up in the locations where they compete best, and that would happen, of course, then what you're monitoring is the extent of competition as it's happening right there and then, and you would rightly conclude that each species is the best competitor. But what you can't do then is predict what will happen when you remove competitors. And that's because when you fit your models, species are confined to their realized niches. But when you're trying to predict what will happen to species when you remove competitors, you have to know about the fundamental niche. And there's nothing in that model that gives me any information about the fundamental niches of the species. And so really, that is my conclusion. Thank you. On here, it's just, it means, sorry, so what does my model say will happen? What does my model predict will be the increase in population size when I remove competitors? and I divide that by the observed. How much do I observe population size increasing when I compare my mixture plots to my monoculture plots?
So going down means
I underpredict.
Tend to be really low in population size, and in fact, you observe them to be really big in population size.
Well, it's the change. When I remove competitors, I predict I won't see a large change, perhaps just a 1 to 2-fold change, yeah? But what I observe is maybe an 8-fold increase. And hence, I haven't predicted accurately what will happen. I've underpredicted the response to the removal of competitors. That's what it is, okay? It's a prediction of how well can I predict how well will these species respond to the removal of competition? And the answer is, I can't predict it very well at all. Yeah. Thank you for that. Okay?
Yeah, I think we're going to have to move on.
Okay, thanks.
Thank you, Lindsay.
Oh,
I don't know.
it looks 4:3 to me.
Our next speaker is Brian Steidiger, and he's going to talk about variability and potential to exploit different organic phosphorus compounds among tropical montane tree seedlings.
All right, thank you. So just a brief run through what I'm going to be talking about today. I'm going to do a sort of a basic primer of niche partitioning and competitive exclusion, just a bit of a refresher. Then I want to talk about some hypotheses I have about how niche partitioning may occur among seedlings, or among tree species rather, in regards to soil chemical phosphorus compounds. And then I'm going to describe some experiments I did to test those hypotheses. So just basically, this has been talked about a lot in these community ecology sessions. We deal first with this idea there's a competitive exclusion principle. So if you grow two species or multiple species together that have identical resource requirements, you find that they cannot coexist. One will deterministically exclude all others. So that's usually stated that complete competitors cannot coexist. And in fact, in natural communities, when we find multiple species, what we often see is that they're not complete competitors. Instead, that they've specialized to exploit different fractions of their environment. And one of the classic examples from MacArthur is where you have 5 different species of warbler here, and they're all exploiting different fractions of this total tree crown. And essentially what I'm interested in is whether a similar phenomena of niche partitioning can occur among plant species that can exploit different fractions of a soil chemical gradient, or of a single soil nutrient rather. So to be specific about what I mean by that, if you think about soil phosphorus, it's not this monolith. It exists in a number of different chemical compounds in the soil and mixture. And we have a combination of inorganic phosphate, where plants don't require any enzymes to acquire this directly from the soil, and a variety of organic compounds. And when you have phosphorus bound up in these organic forms, plants have to produce these phosphatase enzymes, which cleave the inorganic phosphorus from the organic complex so that they can then use it to fuel their growth. And what's interesting is that we can separate these into different functional groups on the basis of their chemistry, but also on the basis of the enzymes that plants require before they can cleave the inorganic phosphate out of the molecule. So for example, glucose phosphate is a simple monoester, but plants would require a phosphomonoesterase to get at the phosphate. RNA molecules are diesters, so a plant would require both a monoesterase and a diesterase enzyme to exploit that form of phosphorus. And then we have these 6-membered carbon rings where each carbon is attached to a phosphorus molecule, and these are called inositol phosphates. And what we find here is that plants require both a phosphomonoesterase and a series of special phytase enzymes in order to exploit this phosphorus. So if we imagine we've got a lot of different plants that are growing in an environment, we can think that the fraction of this soil chemical mixture that they can exploit is going to be dependent on what enzymes they have, or what enzymes they're expressing. And moreover, we have some a priori expectations that plants that belong to different functional groups or phosphorus acquisition strategies should differ in the expression of these enzymes. So I'm going to talk first about the two major functional groups of mycorrhizal fungi. And the first, most plants belong to the arbuscular mycorrhizal group. So they're associating with fungi that's thought to have sort of a low ability to exploit inorganic phosphorus. And a lot of that is based on the fact that all arbuscular mycorrhizal fungi have evolved from the Glomera mycota hundreds of millions of years ago, and they never had a free-living saprotrophic form that had to adapt enzymes for breaking down organic molecules. By contrast, some plants associate with the ectomycorrhizal fungi, and ectomycorrhizal fungi represent several more recent evolutionary convergences onto this symbiotic life form, but they're derived from these free-living saprotrophic fungi. And as a result, it's thought that they retain a lot of the enzymatic ability of their saprotrophic ancestors, and so they should have the ability to exploit organic compounds, phosphorus and nitrogen. However, I should mention that in studies that tend to support the idea that they have lots of enzyme ability, it seems to be dependent on the ectomycorrhizal species. And then finally, we've got non-mycorrhizal plants. And the ones I'm interested in particular are pretty interesting. They're plants of the family Proteaceae. And these guys form no fungal partnership, but they have this really interesting root morphology, where they form these cluster roots, which are these sort of compound clusters of lateral rootlets. And they use these to intensively mine soil for soil phosphorus and other nutrients. And because they have sort of this really intensive phosphorus mining strategy, I had hypothesized that they're probably adapted to exploit a large fraction of the total phosphorus pool, which would include organic molecules. So my basic question was whether plant species with different phosphorus acquisition strategies differ in their ability to exploit different organic phosphorus compounds. The idea being is that if we can show that they have different abilities to exploit these organic phosphorus compounds, we can demonstrate that they have the potential to coexist via niche partitioning, where each is exploiting a different fraction of that total phosphorus pool. And I had a predicted rank order from highest to lowest ability to exploit organic phosphorus compounds that goes arbuscular mycorrhizal plants, ectomycorrhizal plants, and those non-mycorrhizal plants that form the cluster roots. And so if you want to test these hypotheses, you have to go to a place where you can find all these different functional groups coexisting. And I went to Western Panama, the Fortuna Forest Reserve, which is a tropical montane rainforest. It's a site managed by the Smithsonian Institute. And my advisor at the time, Jim Dalling, has a plot network set up there. So we knew that where these plant species were and that they were coexisting. And specifically, what I looked at were, I was interested in focal species from all these different functional groups. So I looked at two different arbuscular mycorrhizal species, one ectomycorrhizal tree species, and one non-mycorrhizal tree species that forms cluster roots. And I conducted two experiments on these, and I'm going to start off with experiment one. So the first thing I wanted to know is whether these guys differ in their enzymatic expression patterns in terms of the enzymes that can break down organic phosphorus. So you take a cohort of seedlings that are growing in the field, you excise their roots, you expose the root sections to enzyme substrate, and you measure the release of product. And from this, you can quantify the activity of different phosphatase enzymes. And in particular, the ones that, so I wanted to do all of the enzymes required to break down all of these forms. But the ones that worked out were the assays of phosphomonoesterase and diesterase. So these are the enzymes that would be required to break down, for example, glucose phosphate and RNA phosphate, and cleave that inorganic phosphate from the organic complexes. And I've gone ahead, on the x-axis here, I've got the tree species, or the seedling species, laid out in the order with which the rank order that I expect them to be able to exploit organic phosphorus. So I'm predicting that you're going to find that ectomycorrhizal plants have a greater ability to, greater enzyme activity than arbuscular mycorrhizal plants, and that non-mycorrhizal plants have the greatest ability. And in terms of phosphomonoesterase, that enzyme that's involved in breaking down, it's at least implicated in all the forms I showed you on that earlier slide. That's essentially what we do find. Oreamunia, the ectomycorrhizal tree species, has around twice the enzyme activity as the arbuscular mycorrhizal ones, although this is only marginally significant. But most markedly what we find is that the non-mycorrhizal tree species has much greater enzyme ability. And when I conducted this test, I was able to assay separately root clusters, so those were those compound root bundles, and find roots from my non-mycorrhizal plant. And in the monoesterase assay, what we found is that the root clusters have markedly greater phosphatase activity. And we did the same thing for phosphodiesterase, and we get a similar result. I'll say there's two main differences here. The first is that whereas in the previous assay we found that the ectomycorrhizal species had greater phosphatase activity, when in diesters, the ectomycorrhizal species actually has the lowest diesterase activity. But similar to the previous assay, we find that the non-mycorrhizal tree species has the highest activity of phosphodiesterase. However, we don't see that same difference between root clusters and fine roots. So basically the take-home point from this slide is that if you were going to grow these guys together, you would expect that the non-mycorrhizal tree species would have a greater access to the organic phosphorus fraction of a total phosphorus pool. So in addition to the enzyme assays, I took cohorts of seedlings from the field and I transferred them to soilless media, and then I gave them all the resources they need to survive, but I provided phosphorus exclusively in one of four forms, either an inorganic phosphate, glucose phosphate, RNA phosphate, or an inositol phosphate. I apologize for the low resolution here. This is a bit of a Mac to PC error. But we looked at different performance measures to see whether these plants differed and how well they grew when they're limited to these different organic phosphorus compounds. And the big take-home point here is that we found in all of our performance measures significant species treatment interactions. So this indicates that the species were reacting differently to the different organic phosphorus compounds. Now, I'm not going to go through I'm only going to go through two of these measures from our experiment too, but they're the ones that I think help me tell the story I want to tell. So on the x-axis, once again, we have the tree species. And on the y-axis, I have a legend that's showing us the different forms of phosphorus in our experiment. And we're going to look first at the foliar phosphorus of our seedlings. And what we find is that in all of our tree species, that we have increased foliar phosphorus relative to the non-phosphorus controls in the RNA, glucose, and inorganic phosphate treatment. So that indicates that they're probably able to take up phosphorus from all of those sources. However, interestingly, the non-mycorrhizal species is the only one that has a significant increase relative to the control in the inositol phosphorus treatment. If we then compare the change in leaf area, so this is leaf area from the beginning of the experiment to the end, we find some other interesting results. First, we find that in the arbuscular mycorrhizal tree species, we only saw increased leaf area in the phosphate treatment. We have a strange negative reaction of Oreamunia leaf area to the RNA treatment. So I'm not sure why that occurred, but it was. I've got an hypothesis there, but I'll leave it at that for the time being. But the most interesting result we have here is that in the non-mycorrhizal plant species, we have almost 3 times increase in leaf area in the inositol phosphate treatment relative to the control. So what we conclude from that experiment is that Rupala montana, this non-mycorrhizal tree species that forms cluster roots, seems to have a unique ability to exploit inositol phosphorus relative to our mycorrhizal tree species. It markedly increased leaf area and significantly higher foliar phosphorus in this treatment. It also showed greater activity of the requisite phosphomonoesterase enzymes that are involved in the breakdown of inositol phosphates. And by contrast, we didn't seem to find any support for the idea that the ectomycorrhizal and arbuscular mycorrhizal tree species differed in their ability to support growth. Or we didn't find any, rather, support for the idea that the ectomycorrhizal tree species had a greater ability to support growth when limited to organic phosphorus. The only big difference there we saw is that the ectomycorrhizas seem to have a really adverse effect when they're limited to RNA phosphorus. So all of this sort of points towards a potential for soil phosphorus partitioning to play sort of a coarse role in niche partitioning, that is, between non-mycorrhizal and mycorrhizal tree species. And it also supports the idea that if we're just looking at the enzyme activity, so just the enzymes of ectomycorrhizal trees, we might conclude that ectomycorrhizal trees should outperform arbuscular mycorrhizal trees when they're limited to organic phosphorus. But our experiment doesn't support that. So I'd like to thank co-authors, Ben Turner, Jim Dawing, and Adriana Corrales, and a bunch of people who helped out with this, and I'd be happy to take some questions.
Time for one question.
Are your mycorrhizal species obligatory mycorrhizal, or they are facultative mycorrhizal?
So I don't know. The ones I had from the field, we did do mycorrhizal colonization assays, and they all were colonized by mycorrhizal fungi.
But you know,
I don't know how well they would grow in a totally mycorrhizal-free environment.
Okay. I'm afraid we'll have to leave more questions until after the session. Thank you very much. Is that the right?
Okay, no,
She's going to work on that.
soon.
She's setting that.
All right, our next speaker is Javier Puig. He is talking about transgenerational effects on plant competition.
Okay, so hello everyone. Thank you for coming. I'm going to talk about transgenerational effects, but don't run away. Okay, I know that is quite a scary name, these transgenerational effects, but don't worry, you are very familiar with this term, you will see. So, because you all know this sentence, right? You are what you eat. This is super common, right? So if we have two identical twins or two clones, and we grow them in super different conditions, after a while, they're going to be quite different, right? They are going to be phenotypically different. Okay, but what if I tell you this? You are what your parents ate. Okay, the first one is basic. It's basic epigenetics, but the second one is what we call a transgenerational effect. But let's explain it in a more ecological way that maybe is better. In nature, we found some phenotypic variation within the populations, right? So although they are the same species, there are some differences. So why are these differences? They can be because they are genetical. They have some difference in the genome. Okay, there's some difference in the DNA, or it can be because if in case of that they have the same genotype, it can be because they were grown in different environmental conditions. So this epigenetic variation is the phenotypic variation caused by an external or environmental factor. Okay, this epigenetic variation doesn't change the DNA, only it has some mechanism that change the gene expression, okay, but not the DNA. Okay, and these epigenetic variations can operate over short periods, for example, within the individual lives. This is normally what we call plasticity, but also it can it may become heritable for several generations. So in this situation, we call it parental, just for one generation, or transgenerational effect for several, so for more than one generation. Okay, and why we say that epigenetic variation is ecologically and evolutionarily important? It's because this change can affect, again, the ecological interactions, and is one of the main factors that for short-term adaptation. So these transgenerational effects has been increasingly demonstrated in plants that suffer different abiotic conditions, but there is not too much about biotic conditions, okay? Because competition is the main driver of the plant community assembly, that's why we ran an experiment in order to try to test if there is some transgenerational effects in plant competition. Okay, so this is our first objective. See if there is some, okay? If this changes in the parental, these different competitive interactions in the parental generation is changing the offspring, okay? So basically we will see these changes in the early development of the offspring. So we will measure in the germination, the growth, and also some resource use acquisition strategy of the offspring. Because we only ran one generation, maybe it's better to say parental effects, okay? But then, in any way, this is the experimental design. Okay, this is our species, our target species, Ataraxacum brevicorniculatum. Okay, it's an apomictic species. So this means that it makes clones of itself, okay, through seeds, okay? It's in order to control that there is not contamination in the genotype with some pollen or something like that. So it's an apomictic species. We grow these clones in different, in competition with six individuals of different 11 species. That they differ, these species differ in their competitive ability. So we more or less characterize the competition that our target species suffer. So we have now the parental environment with a gradient of competitive strength. And then we took seeds from this experiment, from this parental environment, and we grow them in a non-competition and all the same conditions. Okay, we grow them, and for half of the experiment, we add the methylation agent. It's a chemical called azacetidine, I don't know how to pronounce it, azacetidine, whatever. So this removes the epigenetic effect. So it's kind of reset of the plant, okay? They forget where were their parents. Okay, and then we measure the germination rate, the growth rate, and some functional traits above ground and below ground in order to know which is the resource use acquisition strategy of the plant. If it's more acquisitive, so it's fast growing and productive, or is conservative, so it's slow and persistent. Okay, so now here are the results. We found a parental effect, okay? But also it's important to take in account that seed mass is affecting as well these early development traits. Yeah, maybe it's not, well, okay. And let's start with the germination. We don't see any difference in final germination rate, so all of the offspring germinate the same, so there is not difference, but there is difference in the timing. Okay, offspring that were that the parents were in a stronger competition, they germinate earlier than the ones that were with less competition. About just one thing to take in account, all the graphs, the thing that is shown in the graph is the effect of the parental, this transgenerational effect, this parental effect, and is the first p-value. The second p-value is the effect of seed mass that is not presented but is taken in account as covariable. So then the growth, we saw that plants with parents that were with strong competition, they grow faster, okay? They grow faster. Seed mass as well is important. First p-value, parental effect. Second p-value, seed mass. And importantly, we don't find this relationship in the demethylation effect when we remove this epigenetic effect. So this suggests that is only and this epigenetic effect is driving these growth differences. And when we keep going and we measure the growth rate in the day 42, we see that we still have an effect from the parental effect, but we don't found it for the seed mass, because as you know, Taraxacum seeds are quite small, so the effect of the seed mass is only for the first 2 weeks. And about the resource use acquisition strategy, we saw that offspring that come from a parent that was suffering a lot of competition, they tend to be more conservative. All their traits tend to more conservative traits. And this is a normal pattern for perennial plants, okay? Be more conservative when they are in competition, because it improves your competitive ability. And of course, again, in the demethylated part of the experiment, we don't find this relationship. So in conclusion, and coming back with this nice graph of Postdorf, we see that competitive interactions trigger transgenerational effects, okay? And how are they changing the offspring? They are increasing the germination rate, the germination speed of the offspring. It's increasing the growth rate of the offspring, and the offspring is becoming more conservative. So, okay, if we think of a plant that is going to germinate faster, grow faster, and is going to be more conservative, it's going to change again the ecological, this biotic interaction as well. So it's important, maybe, for example, if we think in this example, maybe the competition effect is going to decrease because the offspring is more prepared to grow in this competition. So basically, like the message to bring home is that the epigenetic variation matters. It's nice to have it in account, and is an important factor for our short-term adaptation. So, thank you very much, and open to questions.
I mean, it's a fascinating study and really elegantly produced. Thank you. But I wanted to ask you about seed mass, because I think in this context it's very interesting. A large seed mass could be seen as an advantage for a very small mass.
Yeah.
Lindsay might disagree with that, but no, I wouldn't disagree with you. So, but you seem to analyze that as a separate thing rather than as a transgenerational effect. Could you comment on that?
Yeah, very good question. The seed mass is considered as well as a maternal effect, okay? Because the mother is the one that is giving you some resources. And I skipped that part because I didn't have time, but we saw that the mothers with more competition, they have less seed mass. Okay, it's understandable. But if we take in account this in the other results, so like, of course, the seed mass, for example, with bigger seed mass, so in that case, it will be in here with less competitive strength, they grow faster as well. So the relationship will be in the other direction because the seed mass. But then the parental effect is giving the other result. So this is why it's interesting to separate the seed mass to the parental effect, because if not, if you take in account all together, you will see just a flat relationship. You know? Thank you.
Question in the back there?
We ran another experiment in order to see if this method is correct and is demethylating as well as germinating, because we spray the chemical. We don't grow the seeds in this chemical, so it's the traditional way to do it. So we spray them, but we measure the amount of DNA methylation, and it's the same. It's the same. There is no significant differences between the traditional one method and this spray method. Yeah, we didn't measure in this experiment, but in a previous methodological experiment, just to control if our method is having the same results, it does. So, thank you.
Okay, I'm afraid we have to leave questions until the more questions until the end. Thank you very much.
Thank you.
It's a PDF, no wonder. Why is that not switching over? There must be some way. Let's see if she can tell me. Try. I wonder if we do this, if it then goes. How do we get the PDF to show up there? All right, okay. Nope. Okay, well, while we're waiting for the technological challenges to be overcome, let me introduce our next speaker. It is Cameron Wagg, and he will be talking about biodiversity promotes ecosystem stability under drought by shifting seasonal productivity. Any minute now.
Perfect.
Okay, thank you.
Great. Thanks a lot. So I'm going to present some of the results from a long-term drought experiment that's a part of the bigger Jena biodiversity experiment, which I'll get to in a minute. So this experiment was motivated, of course, because we see a growing change in our climate. And over the last 100 years, we've seen that there's been an increase in the intensity, frequency, and prolongation of drought in various ecosystems around the globe. So this is a figure from the IPCC 2013 showing the intensity of or increasing intensity of drought in different regions around the globe in brown. Uh-oh, what did I do? Okay. So of course this is a big concern because this means that we might lose species and it will change the composition of the species and how they function and how the ecosystem functions. And more importantly, it can have a dramatic effect on ecosystem services, particularly agriculture. So these bottom pictures are from the severe drought in California that occurred in 2012 that decimated a lot of the agricultural systems. So exploring how and understanding how ecosystems respond to increased drought frequency is of key importance. But we also know over the last 20 years or more that biodiversity is quite important for maintaining the stability of the ecosystem. So this is a figure from Forest Isabel just last year. So we have an increasing number of species here. Oh, I think I have a cursor. And ecosystem stability. So this is the mean through all time of the productivity divided by its variation. So we have greater stability at higher diversity. And this is because we have greater resistance to these climate change and to these extreme climate events and drought events. And drought increased drought resistance is shown here by David Tillman back in the 90s. So to sort of explain this and depict it a bit more, what's going on and what we might visualize this as. So at high diversity, if blue line is our ambient community or there's no drought occurred, and at high diversity we might have high resistance, so it changes very little. But at low diversity it changes a lot, so it would be much less resistant. So that's kind of a hypothesis there. However, if drought has an effect, the system has to recover, so it might recover much faster under high diversity to be more similar to the pre-drought or ambient conditions. Well, at low diversity, it doesn't recover very much, and it's much slower to recover, so there's less recovery and a greater dissimilarity between the ambient and drought conditions. And I have resilience here, and this is just the ratio of how much it has returned to its original state relative to how much it had changed during this environmental perturbation. So this would be the drought conditions, and then we have our recovery. So these two things drive the stability of the system. So if this is years or time or seasons, and the blue line being our ambient or non-perturbed system, and the red is our drought conditions, and over here we have the long-term mean and the variation through time as the arrows, and the mean is the points. So this would indicate how the stability is changing. So if drought has very little effect on the stability of the system, it would indicate that we have a high resistance and potentially high recovery. However, if we have a low resistance, so productivity is suppressed by drought, but we have a high recovery, this would increase the temporal variation in how the system is fluctuating, because it has to recover back to the ambient or to the non-disturbed conditions. So we have this increase in variation, but also at the same time, this will decrease the productivity of the system. So a third scenario could be that the system is destabilized by drought because it's simply strongly suppressing the productivity. So we have a poor resistance, but also a poor recovery. So the overall productivity through time is strongly decreased, but it might not necessarily increase how the system is varying and fluctuating through time. So those are three different potential scenarios that we can think of how drought might destabilize a system. So with that as our sort of conceptual background, we had two key main hypotheses for this experiment. So how does the recurrent seasonal droughts over the long term affect the functioning of the system? And secondly, exactly how does greater species diversity stabilize the productivity of these systems? So this was set up within the Jena experiment. So this is our field site in central eastern Germany in Jena. Some of you may know it. And we have 80 main plots, so those are these big plots here. And this was set up in 2002, so we're celebrating our 15th year this year. And each of these big plots has a different species richness gradient, so 1, 2, 4, 8, 16, or 60 species. And within each of these main plots, since 2008, every summer we would set up these rainout shelters over two 1 by 1 meter subplots. So a drought plot, drought treatment, and an ambient treatment. And we would, this ambient treatment or control treatment, every time it rained, we would collect the water and add it back to the plot. So those were our two treatments. So this is showing over the 6 years where we collected the data from these rainout shelters, and this is what they actually look like. So the blue bars are the mean precipitation per month, and the red bars are those 6 weeks in the summer when we set up these roofs. So we set them up in the middle of July for 6 weeks every summer. And overall, this reduces the amount of summer precipitation that the drought treatment receives by about 40%. So what we find is actually maybe not what you might expect. So over these 6 years, in the summer, the drought overall depresses the productivity, which is maybe not surprising, but there's no influence of species diversity interaction with these drought treatments. So the drought is the solid points in line. But what's really surprising is that in the spring, following productivity, we actually have a much steeper diversity productivity slope. So that means that at this higher diversity in the spring, these communities are more productive if they had experienced the drought the previous summer. This is weird, right? So it was sort of surprised us. But if we look at the annual, so the sum of how they're functioning at a broader time scale, so at the annual scale, we see there's a weak effect, but we see there's still maybe a small negative effect at low diversity and a small positive effect at high diversity. So there's something happening here. And if we conceptualize this in terms of stability, if we just think of how things are varying across seasons, we see that diversity has a much shallower relationship with stability compared to the ambient, the open plots and dotted line. So this is really telling us that across seasons, these more diverse communities are varying much more among seasons. But then if we look at the annual productivity, so the broader scale, the broader time scale, there's absolutely no effect we observe no effect of how these communities are varying through time. So the effect of drought disappears. So we can explain this best conceptually. So what's happening at the high diversity is we do have higher resistance, but we have this compensatory recovery the following spring that compensates for these losses in productivity, so that the long-term annual mean or the annual mean remains unchanged. But the variation among seasons has increased greatly. While opposingly at low diversity, we have a strong depression in the productivity, but the variation remains kind of the same. So they're still varying at a low level among the seasons, and they've just sort of been depressed in productivity to a lower state of functioning. So to try and explain these patterns a little bit further, we started to look at phylogenetic diversity and functional trait diversity to see how the communities have changed through time. So these sort of reassembly filtering effects. And at low diversity, we see that the community-weighted, so functional diversity and functional trait diversity, or phylogenetic diversity and functional trait diversity go down over time under drought, which is perhaps not surprising. But if we look at the community-weighted mean in some of these traits, we see at low diversity, it seems to be favoring the persistence of smaller species, perhaps because of drought resistance. So we have that lower productivity at low diversity. But at higher diversity, the opposite seems to be happening. We seem to be favoring larger, taller species. And perhaps this might be due to some sort of limiting resource competition. We're not really sure yet, so maybe there's some ideas. And with regards to this compensatory recovery that we see at higher diversity levels, I think this can be best explained that during the summer drought, there's very little activity in the soil microbiome to effectively mineralize all the resources there. So there's less of a resource pool that's being utilized during the summer. While in the spring, there's, because of this underutilization of the resource pool or mineralization, there's more that can be mineralized and flushed out of the soil into the system to increase this productivity. So that could be one way in that we get this compensatory recovery at higher diversity. So to summarize in the end, biodiversity can stabilize ecosystem productivity under recurrent droughts because of increasing resistance. So it's nice that we can see in our system and confirm the results that we've seen in other systems. However, if we look at shorter timescales, we see a different pattern, and this increased variation among seasons at higher diversity is actually the stabilizing effect along the broader scale because of this compensatory recovery. So I think there's a lot of potential left that we still have to explore. So what are the ecological and community level consequences of this altered seasonal productivity and how the system functions? And that's where we're at the moment with this experiment. So I'd like to thank all of my collaborators and people that have worked on the experiment over the years, and of course, all the technicians and management team that maintained the experiment. And of course, thanks for your attention, and look forward to your questions.
So on one of the last slides that you showed where you tried to reason why you have the spring over compensation in the high non-diversity spots, you could use the same reasoning for
This one.
Yes. And one, yes. I mean, I can see that, but this could also hold true for the species, for the low biodiversity plots, right? Because also there, the microbiome, the soil microbiome will be
Yeah.
less productive during those drought season, and there would also be then more stuff left that could be remineralized next spring. So why does it not happen there to the same extent?
Yeah, that's a good question. I think so. This is all very speculative to begin with, that we haven't actually measured yet. But, or I'm not sure if we will. But I think you're right. But also, there's less litter being put into the soil. So at higher diversity, you have much more biomass and much more dead material and root turnover that's happening. So I think there's just a greater pool of organic matter in the soil. So that could be one reason why maybe that's not an explanation for the low diversity. But
Will you investigate
yeah.
also the biodiversity in your soil microbiome for the different plots?
So that was measured previously, and we have no plans for it. So at the moment we're looking more. We hope to delve more into the community aspects, but I think really a lot of the underlying mechanisms are these plant physiological soil interactions that's happening. So, yeah.
I wonder, do you have high mortality after the drought?
Not really. So it's more. So the first few years, it didn't really seem that there was a lot of change in the realized species richness between the plots. But in the last, after 5-6 years, we see this becomes quite strong. So it took some time. It's not immediate.
Because I was thinking that maybe you have less communities after the drought, and this is why you have higher recovery in the high diversity treatment. It's because you have lower number of competing individuals, so individuals can be bigger, and this is why you have higher productivity in high diverse environments.
Yeah. I didn't think about that. Yeah, I should mention they're all sort of annual plants, so they. I think the number of individuals or plants species that disappear after the drought it's not as strong. I mean, it seems to have taken some time, but that's a good thought. Yeah.
Okay, well, I believe we have to move on. Thank you very much. Okay, there's our next page. Let's see, are you wide or?
Sorry?
Is your screen wide or not?
I have no idea.
Okay. I didn't know that. She will take care of that. Yeah, that looks like that's fine. It's not cutting anything off, is it? Okay, great. All right, our next speaker is Alexis Sinodinos, and he is talking about forecasting and preventing irreversible woody encroachment in savannas.
Yeah. So, hi everyone. Thanks for being here. So I'll be presenting to you a study where we basically modeled savanna dynamics, and we tried to find early warnings of woody encroachment and essentially test whether they were early enough for us to prevent encroachment. So as I've noticed, not many savanna talks in the sessions. When I refer to savannas, basically, I talk about mixed tree-grass biomes, very simply. And the phenomenon of woody encroachment essentially describes the phenomenon whereby the removal of grass cover allows woody vegetation, which aggressively colonizes the landscape, to dominate and basically create patches of shrub cover, woody cover, and bare soil. So obviously grazing is a driver of this process, because grazing removes grass cover, so it kind of creates a double impact by removing direct competition for resources, but also it weakens the fire disturbance. So in savannas, fires require grasses to burn, of course, but they delay the recruitment phase of trees into adulthood. So removing grass cover creates this double impact and speeds up the process of encroachment, basically. And when I talk about reversible encroachment, essentially I'm talking about hysteresis. So if we observe that encroachment is already occurring, an irreversible process would mean that if we return, for example, our grazing, or if we return the system to the pre-encroachment conditions, it will not actually restore the system to its previous state. So it's basically a transition to a different state that can't be reversed by simple restoration processes. And exactly for such transitions, there's a whole theory of early warning signals that's been developed, and essentially it just looks at spatial or temporal patterns in the dynamics of the system, which can tell us that the system is actually approaching such a transition. And a typical type of warning signals are these critical slowing down signals, which essentially tell us that the system is becoming less resilient because it's slower to respond from a perturbation. So if we push it away from the steady state and its response is slow, it means that the system is not very resilient and probably approaching such a transition. So just to quickly show you the model, so we have a model of grass and tree. Is this the, no. Grass and tree interactions, is this the one? Yeah. So grasses and trees. And I'm just showing grazing here. So if we remove grasses from the equation, it has the impact of reducing direct competition, but also weakening the fire control on tree recoupment. So we asked 3 questions. A, do we have early warning signals in our simulated savanna system? B, can we find signals that distinguish between reversible and irreversible encroachment? And C, if we find such warning signals, do they actually show up early enough for us to do anything about it? So this is, okay, so here essentially I'm just showing you the fact that we found these warning signals. So autocorrelation and standard deviation are typical signals of slowing down, and autocorrelation and standard deviation in a leading variable such as tree cover. So if we look at the grazing gradient and we look at low grazing, for example, we only have grasses because trees cannot survive. And if we look at the other extreme of grazing gradient, we only have a single state of high woodland, savanna. But here in between, we basically see the two, well, the 2 alternative states of woodland, encroached savanna, and open savanna or grassland. And just to show you, to illustrate that, for example, if we started with a high initial tree cover and we increased grazing gradually, suddenly we would have the creation of the alternative state. So suddenly, from where we thought we only had grasslands, a tipping point is crossed, and an alternative state is created. And this is where normally, I mean, we see the warning signal picking up. This is where normally warning signals have been applied. But we were more interested in actually using them for management purposes. So we asked a different question. We said, let's assume we have, within this area of biostability, we look at the different grazing values, and we simulate different initial tree cover values. And essentially the white region is the grassland basin of attraction, so all the simulations converge to the grassland state, and in the black region all the simulations converge to the woodland state. So essentially what we wanted to ask is, if we start in the grassland basin of attraction, so somewhere here kind of close to the transition point, and induce a short-term perturbation. So if we increase grazing for a short time and then bring grazing back to its original value, will this cause a transition, and will we be able to see this in a warning signal? And will this warning signal be able to tell us whether this encroachment is simply short-term and will subside, or whether this is a long-term transition to the alternative state? So we use these 6 grazing values, and we induced 10 different scenarios of grazing increases for each value. So we increased grazing by 10%, 20%, all the way to 100%, so basically doubled it for all these scenarios. So essentially we created 60 scenarios of heavy grazing treatments. On the y-axis here we have the likelihood of transition basically to the encroached state. So out of all the simulation iterations, I think there were 100 simulation iterations, what proportion of the simulations converged to the encroached state despite the return of grazing to its initial value? So which scenarios led to a long-term irreversible encroachment? And more importantly, we plotted this against 3 different early warning signals, so autocorrelation, standard deviation, and the change in tree cover from the beginning during this 30-year heavy grazing treatment. And I mean, it was kind of encouraging to see that there was a correlation between the likelihood of transition and the warning signals, but the problem is with this kind of result, it doesn't mean much from a management perspective. I mean, if you have an autocorrelation of 0.4, what does this mean if you don't have another 60 points to plot and the y-axis already given? I mean, if you don't know the output, it doesn't really help you forecast much. But by actually looking at the temporal dynamics of all these warning signals, I actually came across an interesting pattern when looking at the standard deviation of tree cover or woody cover. And what I noticed is that woody vegetation standard deviation increased over time in the high-risk cases. So here, for example, we plotted a 5-year delta ST against a 10-year delta ST. So this is the difference between the last 5 years and the first 5 years, and the difference between the 10 last years and the 10 first years. It's just a measure to show how standard deviation increased over time, basically. And well, for the cases that had a high risk of encroachment, it predicted 100%. I mean, it found 100% of the cases, and for the cases that had a 50 to 75% encroachment, irreversible encroachment risk, it picked up 5 out of 7 cases. So basically, this is the indicator that we're looking for. Simply a positive value, so increase over time, meant that we are in a high-risk scenario. And the really cool thing, at least from my perspective, is that we don't require any prior knowledge of the system, of the underlying dynamics, of what our initial grazing value was, what the initial tree cover. This is just observing the 30-year time series of woody cover and seeing, is standard deviation increasing over time? Is it not? And this actually was a pretty good predictor of what was happening. So shrub encroachment or wood encroachment data are quite hard to find, and we found this from another study that the authors were very kind in providing us with. We found this so they had 6 different plots of grazed and ungrazed treatments with different fire return intervals, and this is basically the shrub cover data from the experiments. And we used our delta ST warning signal up to the point, for this time series it's not so obvious, but for the rest, the encroachment hasn't actually started to develop. So there's no warning here that encroachment will occur. And the warning signal actually was positive for all the 4 cases that eventually became encroached. So these 4 cases became encroached, and when I mean encroached, I mean there was an abrupt transition and a reversible transition. The one case, there's one time series, I don't know what happened here, nothing happened, we can ignore it. But the one case where encroachment was just really gradual and essentially reversible, the warning signal correctly predicted that this was not a case of irreversible encroachment. And again, just by looking at, the time series up to here, there's not much difference between them. So this gave us some, this was some type of success in a way. So this answers kind of the second question. So yes, we did find a warning signal that can be used as a forecasting tool and can distinguish between the reversible and the irreversible encroachment cases. And then the last question I asked was, does this show up early enough for us to do anything about it? If we can't do anything about it, it's not a really useful tool. So what we did is we selected I'll show you two examples here, cases where the warning signal showed up and there was a high, a 90% likelihood of encroachment to the alternative state. So at the end of the heavy grazing treatment, so at the end of these 30 years of increased grazing, we implemented some management treatment, so to say. So what we did is basically we reduced grazing by a certain percentage, and we did this for a certain amount of years, just to see whether it was actually avertible, so to say, the encroachment. And the results show it was, but still, a 20-year reduction of about 50% of grazing is something that no manager really wants to do. So this shows that even with a forecasting tool that works quite well, we really need to do a lot of work in order to prevent encroachment from unfolding. So, yeah, the question is, can this signal, can this indicator be validated on more data? And because it's, in terms of its what we require in order to apply it, we just need to monitor tree cover or woody cover, which is fairly simple to do and quite cheap. And if we can actually find more data to validate it on and maybe refine it, then I believe this could be a really, really powerful management tool in order to prevent such a process of catastrophic process of encroachment. So yeah, that was it. Thank you for your attention, and hopefully you have some questions for me.
Yeah, I don't know much about savannas at all, but it's interesting that when you say you start with a grassland, you bring in a bunch of animals and you turn it into a woodland, which is completely opposite to what you see in Europe.
Yeah, I know this.
You take the grazers away and then you're going to get into it.
Yeah.
So what's that? What is that from? Is it that the trees in savanna regions are completely herbivore proofed, or
well, I mean, there's a lot of explanations.
well,
The idea is,
couple of them.
well, the idea is that trees and grasses compete for water, and basically the grazers that eat grasses don't really bother trees that much. You have browsers that affect trees, like the impala, whatever. They go and chew on the little seeds of the acacias, but grazers, they just go for the grasses. So
they just don't eat the tree seedlings
no,
at all.
no.
Okay.
I mean, there's some
Yes,
mixed
that's
feeders,
different
but yeah,
stuff.
that's the difference. Yeah.
Thank you.
Is that a question?
How much is your indicator depending on the model you start with? Because of course that model has assumption, and we could discuss for a long time because there's a lot of literature in savannas,
Mhm.
like you're using grazing, not fire, okay,
Mhm.
whatever. So how much? Because I think that the model could be, challenged, and we could discuss long. But as you seem to have an indicator that you derive from a model, but then you use with data, and you even imply that it could be used from management. How much does this indicator depend on the model you started with? And if not, why?
I would say it doesn't depend on the model, because essentially all it I use the model to develop my ideas, basically, and in the end I found an indicator which just observes a time series. And actually the dynamics in the model were actually very gradual. So the encroachment that occurred was very, very slow, even in the high-risk cases. And what you see normally in nature is these abrupt shifts. So even without having such abrupt shifts, the model was actually more conservative than what happens in nature, and still it managed to, it picked up these indicators. And as I said, it just measures variability over time. So it doesn't have to do much with, of course it's based on the model, but I think, and based on this result as well, and we also did a sensitivity analysis for different durations and when the indicator showed up, it was successful. If it didn't show up, there was no risk of encroachment. Basically, yeah.
I think to answer my question, you should try with the model output of another model, which has different mechanisms. Because
Yeah, or more data. Yeah,
or of
either way.
course with data,
Yes.
no, but
Yeah,
otherwise, why to use
if you have such outputs, I'd be happy to use them. Yes.
Okay, thank you very much. She'll change it to the. Where'd my mouse go? Oh, maybe it's because she's doing that. Uh-oh. Where'd it go? Oh, there it is. Okay. No,
There we go.
Good. So our next speaker is Adam Kimberly, and he's going to be speaking on the extent of ongoing plant community homogenization following historical grassland reductions depends on habitat type.
Thank you very much. So I'm from the University of Stockholm. I'm doing a postdoc with Sarah Cousins, looking at how landscape change affects plant communities over time and the way different sort of landscape and environmental variables affect the speed at which communities respond to different shifting forces. So for this project, I'm working in with an area just south of Stockholm. It's a small landscape which has a really long history of human activity. I think people have been there for over 4,000 years. And there's lots of historical map data and information on the land use history from the 16th and 17th centuries right up to the 1980s. And now it's a nature reserve, and grazing animals are maintained at a sort of low intensity on these semi-natural grasslands. And this is just one of the oldest maps which shows where this grazing happened in the past. But it's an area that's undergone a lot of change in the past. So by looking at these maps, you can see that the area of grassland declined quite drastically between the 18th century and 1945, and then again between 45 and 81. And that was because of an increased conversion to arable fields and then an abandonment of grassland and forest encroachment onto those abandoned grassland areas. So basically, we're working with a pair of vegetation surveys. The first was carried out in 1997, and then the sites sampled were revisited last year, so a 17-year time period. And we're interested in whether the community composition in these plots are still changing in response to the historical landscape changes. And because they're randomly spread across the landscape in all sorts of different habitat types, you can see whether the rate of different compositional shifts is varying according to the habitat type that's being sampled. So basically, you might expect that the extra shading in the forest habitats might cause a faster rate of change because of the loss of shade-intolerant species, whereas the management that's still ongoing in the grassland areas might buffer some of those changes. So to do this, because there was quite a small sample size of the individual habitats, I grouped them into open, semi-open and closed habitats. It sort of reflects the environmental conditions and the management history that they've undergone, and made it into a bit more manageable analysis. And then just basically investigated the change in diversity metrics and trait composition between the 1997 survey and the 2015 survey. And what we found, this is just the change in species richness between surveys. There was no change in the average species richness of plots between surveys across the whole landscape, but when you split it up into these categories, there was a loss of biodiversity in the closed plots and no change in open or semi-open habitats. So that, I think that sort of fits with what other people have found in forest habitats over time. If they're abandoned, you lose a lot of those shade-intolerant species. And in terms of compositional similarity, this is just a metric which uses the probability of species occurrence to determine whether species have become more likely to occur homogeneously across habitat plots or are split more unevenly across habitat plots. And so you can sum this at the landscape scale to determine whether the plots you've sampled have become more homogeneous or heterogeneous over time. And the overall landscape did become more homogeneous in terms of the species composition. And again, when you split this up, this seems to be driven by a significant shift in compositional heterogeneity in the semi-open and closed plots. But again, the open habitats didn't see any change. And if you look at the individual species scores, so this classifies the significant winners and losers, the species that got more common within the landscape versus the species that became rarer within the landscape. There's a fairly clear trait pattern. Woody species were almost always became more common, a lot of tree species colonization in some areas. Herbaceous annuals very often lost out and became rarer across the landscape. And it was often short species with heavy seeds, which are traits typical of forest specialists, which were the winners. So I think this is a response to the shading in those semi-open, abandoned grasslands and forest habitats driving these changes. And you can see this if you look at the trait composition between different habitat types. So there's a significant increase in the mean height in closed plots, but no change in open and semi-open plots. And the same for woodiness, a higher proportion of woody species in closed plots, but no change in open habitats. And interestingly, the annual proportion of annual species actually decreased in all plots. So I think this is sort of fits in with the idea that perennial species are quite good at holding on following unfavourable landscape change, but the annual species are being lost more rapidly, even where the management is being maintained. So it looks to me like the open habitats are remaining fairly resilient to that landscape scale reduction in grassland amount, but that might be because actually this landscape still contains a reasonably high proportion of grassland habitat. So the sort of connectivity losses associated with that might not be enough to cause any real damage. But the increase in woodland cover seems to have allowed forest species to start to colonise some of the unmanaged habitat types and where the grazing isn't present. Which is interesting because that change is happening at a greater rate in those habitats. It suggests that once the forest species are colonising those areas, the loss of grassland species because of the increased shading and the environmental change might sort of feed back into that to increase the rate over time. And the other interesting thing, I think, is that it suggests that actually the colonisation processes and the extinction processes following landscape scale change aren't the same, and they aren't necessarily following the same linear progression over time.
So do you think you would find a similar, have you looked at this habitat, does it match what's happening in other parts?
We are looking at a longer time series of landscape change data in a different part of Sweden, where hopefully we'll be able to, I mean, it's not quite it's more of a space for time thing, but hopefully we'll be able to sort of track the changes a bit more closely. I think it fits with what other people have found in abandoned grasslands in other parts of Northern Europe, yeah.
And then my other question is, do you have any idea what the vegetation was like before having human habitation?
Not,
I know
no,
that's a long time ago.
no. But the grasslands have been managed for a very, very long time. So I suspect it's unlikely that there's any sort of lingering effect from there? I mean, you're talking over 400 years of grazing on some of these.
I'm just thinking about, that band, if you're going across the globe there, you get boreal forest all the way across.
Yeah.
Okay, well, we'll move on to the next talk, which actually puts us right on schedule. Thank you. Our next speaker is Gianluca. He's speaking about synergistic effects of dispersal and environmental heterogeneity on the replacement and nestiness components of beta vectors.
Okay, so hello everybody. Today I'll give you just a brief overview about this research on the mechanisms shaping nestiness and replacement in metacommunities. So overall, we can say that the consolidation of the metacommunity concept during the last decade has increased awareness about the interaction between local environmental conditions, here represented by different patch colors, and dispersal from the regional pool on biodiversity patterns across several spatial scales. In reality, though, most empirical metacommunity studies have only focused on total beta diversity patterns, that is, patterns of total variation in species composition among habitat patches. Whereas in reality, beta diversity can be separated in two distinct components, nestiness and replacement. I illustrate here these two scenarios by representing two metacommunities, one with a pattern of idealized nestiness and the other with a pattern of idealized replacement. So these two metacommunities are composed by 8 local communities, and each colored circle is a different species. So that in the first metacommunity, the species poor sites form proper subsets of species richer sites. Whereas in the second metacommunity, we have a pattern of in which different species actually replace each other from site to site along environmental or spatial gradients. Now, depending on the relative importance of each of these components, different conservation strategies are required in order to protect the regional species pool. For example, a dominance of nestiness in a metacommunity would suggest that we can just preserve a few sites with high species richness to contemplate all species present in the metacommunity, while a dominance of replacement would demand the conservation of multiple sites along environmental or spatial gradients in order to protect the regional species pool. So a key question is, what are then the mechanisms shaping each component of beta diversity? Well, there is some indication that spatial isolation actually increases the relative importance of nestiness over replacement. But those suggestions come from observational studies that have used spatial distance as a proxy for dispersal rates, rather than linking actual dispersal to metacommunity assembly processes. Unfortunately, this has intrinsic limitations because the pure spatial signal on diversity patterns can result either from dispersal limitation per se or from unmeasured environmental variables that are spatially structured. So in order to overcome those potential limitations, we have revisited here a zooplankton metacommunity experiment in which nutrient additions and dispersal rates were manipulated independently. We choose to work with these factors because Increasing fragmentation and eutrophication are global trends that are affecting biodiversity for different organism groups, both in aquatic and terrestrial ecosystems. Now, eutrophication can result in directional species losses and affect beta diversity mainly via nastiness, or alternatively, eutrophication can also result in high species replacement if different species have different tolerance to eutrophication. Yet, it's not entirely clear how dispersal would interact with eutrophication to determine this balance between nastiness and turnover in metacommunities. So to explore this, we performed this experiment in which we had 2 nutrient levels, that is basically high and low nutrients, and 3 dispersal levels, so no dispersal at all, low dispersal, or high dispersal. We then used the Baselga approach to calculate nastiness and replacement, and we used Permanova to test how nastiness and replacement changed as a function of nutrient conditions for each dispersal level separately. In a second step, we used PermDisp to assess how nastiness and replacement within each nutrient level, so mimicking here a more homogeneous experimental landscape, is affected by dispersal rates. So to make a long story short, what we found is that in more heterogeneous experimental landscapes, so across the nutrient level, lack of dispersal resulted in beta diversity reflecting only nastiness. This suggests that high nutrient conditions actually removed directionally some species from the regional species pool, and the high nutrient mesocosms were proper subsets of the low nutrient mesocosms. However, increasing dispersal erased completely this nastiness pattern and resulted in beta diversity reflecting only replacement. This suggests that dispersal actually can rescue species from extinction, and at the same time, it allows species to track suitable environmental conditions at the meta-community scale. However, we also found very distinct patterns for each component when we look at more homogeneous landscapes. In homogeneous experimental landscapes, more specifically, we found that the average replacement increased significantly with lack of dispersal. So this is just the opposite pattern as found for more heterogeneous environments. So here we have the average replacement, and the colors are pairwise comparisons across the dispersal treatment. On the other hand, we actually found that the nastiness had just a slight trend to increase with increasing dispersal, but this was not significant. So just to wrap up, we found that the nutrient gradient actually resulted in directional species losses, which can result both from, either from intensive competition in high nutrient conditions or habitat filtering, we don't know. However, high dispersal actually erased this pattern by rescuing species from extinction, and it also resulted in a high species replacement at the meta-community scale. So this actually suggests that in more natural landscapes, considering the same environmental and spatial context, we would expect that more mobile organisms would show a pattern of high species replacement, while less mobile species would tend to present more nastiness. But we also found very distinct patterns in homogenized landscapes, which is actually a likely scenario considering, for example, global environmental change such as widespread eutrophication or global warming that tends to homogenize the entire landscape. Imagine, for example, an entire agricultural landscape that is highly homogenized. So, yeah, that's the general conclusion. I just would like to show here different environmental pressures that are associated with anthropogenic activities, such as urbanization, which will not only change local environmental quality, but it will also change connectivity patterns in the landscape. So these two forces are likely to interact and determine each component of beta diversity in contrasting ways. That's it. I'm happy to take questions. Thank you.
That's a nice experiment. I have just a question about a plot. How can the L square be negative?
Yeah, so this is because, for example, when you use a method such as Permanova or another one that uses Monte Carlo randomization procedures, then you have your observational data that is compared against new models. So in a situation in which your observed data actually fits poorly than a random pattern, your R square will actually be negative. However, this is just interpreted as non-different from zero, that is it's not significant at all. Okay. Thank you.
This will be a more statistical talk, so a bit of a warning.
So if there any more questions for any of the earlier speakers, now might be a moment to take some.
Too close to 5 o'clock, I think, isn't
I
it?
know, well,
Everyone's flagging after a whole day of it.
The title has been simplified a bit. The title has been simplified.
Okay, well, I
yeah,
will read it off the screen for
Yeah.
you then, how's that?
I think it's more
Well,
fitting.
We'll
Yeah.
assume that we won't be breaking down the doors anytime soon. So the final speaker today is Christa, and he's talking about modeling the joint abundance of more plant species pinpoint cover data.
Thank you. I'm going to talk about a case study of dry heathlands. And as you can see on this picture, it's a patchy distribution. There's areas with Calluna, and there's also areas of a grass species, Dischamsa flexuoso. And that will be important in my talk. So why should you model the joint distribution of spatial plant abundance data? If we know a species is present at a position, then the probability that another species present at the same position is reduced. So the plants take up space. And if we use information, then we have more statistical power. And it's also possible to fit ecological models to multivariate abundance data instead of summarizing the variation by ad hoc distance methods. For instance, the break curtains and the whole ordination techniques. So I suggest that these kind of techniques will replace vegetation ordination somewhere in the future. And then we'll also have knowledge on spatial covariance patterns. Plant species are patchy distributed, both large scale among sites and at a small scale within sites. And in this talk, I'll mainly talk about I'll talk about both types of spatial variation actually, both within sites and among sites. Cover at a site is beta distributed. That is a take-home message, if nothing else. That plant species are patch distributed, and then single species cover data are often L or U shaped, especially for the more dominant species. Of course, very rare species do not show a large L or U shaped distribution. This is the beta distribution that has been reparameterized so that you have that the mean of the plant cover at the site is Q, and the variance has a nice form here, and both Q and delta are parameters between 0 and 1. Q measure the mean cover at the site, and delta measure the interplot correlation. This is the single species case. And here's an example of the beta distribution, but I said you can also model a U distribution, which is quite commonly found, for instance in dry heathlands. These are the data I'm going to talk about. It's dry heathland. There's 179 Danish dry heathland sites, and each of them has between 10 and 40 plots. And this is the cover of Calluna vulgaris, the dwarf shrub, Deschamps flexuoso, which is the grass, and then a joint class of other species, because I was focusing on these two dominating species in this case. And the way the cover is measured is that we have a so-called a pinpoint frame that you put into the vegetation. Then you take a thin pin and lower vertically into the vegetation, and then you record the number of species, or the species that touch the pin. And you do that for each of the 16 grid points here. So you have presence-absence data at these 16 grid points for the species that are present in the frame. Yes. And we have worked a lot with the single species case. And remember the cover at the site was beta distributed, and then we had this binomial process at the pin. And then you could assume that the pinpoint data at the single species was beta binomial distributed, and that is actually a good model for the single species pinpoint cover data. And you can see here for Calluna and Deschamps that these are the observed cover at the pin. If you don't take the spatial aggregation into account, so assume it's a binomial, then it's the yellow distribution. And if you take this extra parameter into account, you get a fairly good fit of the observed cover. And this actually plays a tremendous role in the statistical inferences later on that did this extra parameter. We have done that with many species, and it's more or less a good representation of the data for in most cases we've seen. Now we are going to several species and also pinpoint data. And this beta binomial distribution will, in the multidimensional case, be generalized to for n species using the Dirichlet multinomial distribution. It's a standard generalization. So now we have yi are pinpoint hits of species I, and since you can hit more species with a pin than one, the sum of I is may be larger and often is larger than the number of grid points. So imagine that you hit both the grass and Calluna with the same pin, then when you sum them together, you will get more than the number of grid points. In our data set 16. And then we have the relative cover of the species, not the cover, but the relative. It's a small but important distinction, where the relative cover sums up to 1. Cover is defined as the two-dimensional projection of the plant species onto the surface. So actually cover is also expected to be larger than 1 when you sum them together. But in this model, I work with the relative cover. It's for mathematical convenience and generalization, I think. This is what it looks like. It's good we have computers. And before, we had the nice probability that the mean of these are the parameters. And this delta is still the interplot correlation due to spatial aggregation between 0 and 1, but now in the multispecies case. And that is the only way of writing this formula this complicated. That is to get this nice property, because then you can easily fit hierarchical models. Because there you want Latin variables, and often you want the mean to be the Latin variable, and now you have the Latin variable here. So that's very convenient when you later on want to use the model. Okay. I tried the model in a case. So here we have the sites, and it's a Western distribution of but there are plots also over here. And in this demonstration case, you might say, I regressed it only to precipitation. We are currently fitting relatively large structural equation models with these kind of data sets to see what's the effect of different drivers. But in this case, only precipitation, and precipitation is then set, so you can see what is the effect of precipitation on the covers of the different species. In this case, OE3, Kaluna, Deshamsha, and the rest. Estimation, it was a hierarchical Bayesian modeling approach. Mean site cover was modeled by Latin variables. There was this nice transformation. There was the Metropolis-Hastings algorithm in an MCMC. And the statistical inference of the parameters were based on the marginal posterior distribution. And this is some slides how they are reported. And then maybe I should have made graphs of it, but I hope you. So this is the 2.5% percentile, 50% percentile. I cannot see it. Can you see it? I'm sorry. I'll tell you. 2.5 percentile, 50% the median value, and 97.5. So 95% credibility interval between this number and this number. And this is then the intra-site specific spatial variation. There's intra, and later on I'll discuss the between site. We see here that there is variation, and it's larger than 0. We also expect that because it is these dominating species. And if we do not take this spatial aggregation into account, then the statistical inference is biased. It's like pseudo-replication. Because if you have this U-shaped, and if you analyze them without taking this spatial aggregation into account, you assume that you have too many degrees of freedom. And this is a mistake. It's a serious mistake. It makes the p-values much too low compared to the real p-value. And I think it is done on a daily basis in plant ecological communities. And it's reported in journals, and I suggest that we have, there is so depressingly many results reporting cover in the ecological journals where this is not taken into account, and you cannot believe the p-values. They are simply too low. I have some examples that I didn't think I had the time to show, but it's very important. So please, it's beta distributed, and you have to take it into account. It's a parameter, so we can use it for testing different hypotheses. I'll not go more into it, but you can test, so what is the spatial effect of different phenomena. In this case, we were investigating the precipitation on dry heathland, and this is beta. So beta vulgaris. So on Kaluna vulgaris, so that this beta, the effect of precipitation on cover is increasing, and this is parameter measures. So in how many simulations were larger than 0, and this means that 2.5% were less than 0, so it's significant on the 5% level. So the cover of Calluna vulgaris increased with precipitation on the large scale. So among sites. Likewise, this is beta Deschamps flexuoso, the grass, decreased significantly with precipitation. So a relatively high precipitation is correlated with low cover of Deschamps flexuoso and a high cover of Calluna vulgaris. I'm not talking about competition. If you have to talk about competition, you have to have time series. This is only a spatial study. And you have to have time series with permanent plots. So it's a spatial studies and statistical correlation between covers and precipitation. And there was also spatial covariation among the sites. You may expect that from the previous result. I'm not to talk too much about it, but this covariation indicates that the oceanic continental climatic gradient determined the abundance of the two species. Denmark is a small country, so say that this oceanic continental climate gradient is a little bit stretching it, but that's what the data suggests anyway. Conclusions. So the framework allows modeling of the spatial covariance and cover at 2 levels. The local one, which is very, very important for the statistical inferences, and regional among communities of sites. We know from manipulated experiments that drought increased the competitive effect of Deschamps on Calluna. So, and this is kind of in agreement, in concordance with these results. That's what I write here. And it is suggested that this oceanic continental climate gradient determined the relative abundance of the two species. But there are also some effects of soils which may confound these results, and we're currently studying this in these structural equation models that I mentioned before. And then we suggest that maybe we could use the cover ratio of these two species as to monitor the effect of climate change on dry heathland, maybe outside Denmark also. Thank you.
U-shaped business again, why you quite often see that. I didn't fully follow. Sorry, it's the 5 o'clock effect. It's nothing to do with your explanations.
So you have, when you measure cover, and this is a relatively small frame, so it's a half meter by half meter. Yeah. And then sometimes this frame is put in a patch of Calluna vulgaris. And then you put in the pin, you have 16 pins, only Calluna vulgaris. Next time you put the frame into only the grass, and then you have 16 of that, and 0 Calluna. And that gives you the U shape. So, and of course, the larger you make this frame, the less U shape you get. But you have to have really, really large frames to get rid of the U shape in many plant communities, especially in dry heathlands. I hope I didn't say Calluna had a significant effect. It should be precipitation had a. So,
precipitation had a significant
yeah. So this is the model. So.
But the confidence intervals for Calluna are less than 0 and greater than 0.
Yeah, and that's because I'm working with the distribution. It's in a Bayesian context. Yeah. So now I'm not trying to test a null hypothesis. I'm just trying to see whether this beta value is lower or higher than 0.
And in that case, it's...
And 90, I can't see the number. 97% of iterations in the MCMC was larger than 0.
Okay.
So from a Bayesian perspective, this is significant because it's 95% are large, more than 95 are larger than 0.
Yeah,
So, but it's not the 0 test, it's null hypothesis test.
yeah, I was looking at 2.5% column and the fact that that's a negative. But it's
Yeah,
such a small number.
this is a very small number. So yeah, yeah, both of these are it's just that.
Yeah.
It's just tipping right there.
Yeah.
So on the 5% it's significant. On the 2.5% alpha value, it's not